Hello my dear students, welcome to the MS Science Academy. Myself Meenu Srivastava, and today we are going to discuss about the gluconeogenesis. As in the series of the carbohydrate metabolism, we are going to discuss about the gluconeogenesis. What the word gluconeogenesis represent? Gluco means glucose and neo means new and genesis means synthesis or formation. Means the glucose formation from the new substance, and in the biochemistry the new substance means the non-carbohydrate source. So here the gluconeogenesis means the formation of glucose from the non-carbohydrate source. Now we will write the definition. Formation of glucose from non-carbohydrate sources, from non-carbohydrate sources. And what are the sources? These sources are glycerol, lactate, phosphoenolpyruvate, propionate, glycerol, glucogenic amino acid. Okay, so these important compounds. So compounds are here COMP dot means compounds are glycerol, lactate, pyruvate, glycogenic amino acids, propionyl coenzyme A, or you can say the propionate. Okay, so these are the some different compounds which are going to generate the glucose for gluconeogenesis. Okay, so what are the sites? These are the where in the cell which part is primarily involved for the gluconeogenesis. So the cytosol, okay, the main site is the cytosol, but some reactions are proceed in the mitochondria also. Okay, some reactions proceeds in mitochondria. Okay, so mitochondria also having the some parts play some role in the gluconeogenesis, but the main reactions are proceed in the cytosol. Okay, so what are the organs? Mainly organs are involved. The main organs which are involved in the gluconeogenesis, that is the liver, and some extent some part of the kidney like the renal cortex also involved in the gluconeogenesis. Okay, so organ is that liver and kidney. I will write that kidney, renal cortex part. Okay, some up to the some extent, but the main organ is the liver. Okay, so there are so many reactions in the gluconeogenesis, but here we will discuss some specially or the important reaction which are very much important for question for the examination point of view. Okay, so first we will discuss about the reverse glycolysis, which is a very much important for your examination purpose. So first production of the glucose from the non-carbohydrate is the reverse glycolysis. Okay, why we are saying this, the reverse glycolysis? Because as we know that in the glycolysis the glucose is converted into the two molecule of pyruvate. So again we will proceed this reaction that pyruvate will convert into the glucose again. So this is the reverse glycolysis. So this reverse glycolysis also having the some stages. So we will discuss one by one. It include the 3 stages. So in the first stage we will include that the conversion of pyruvate into the phosphoenolpyruvate. Synthesis, conversion of pyruvate into phosphoenolpyruvate. Okay, so here what main reaction is that in this reaction the pyruvate is converted into the phosphoenolpyruvate. Okay, the pyruvate is converted into the oxaloacetic acid. As we know, in the TCA cycle, the pyruvate is converted into the oxaloacetic acid. Okay, so here the pyruvate is converted into the oxaloacetic acid with the help of pyruvate carboxylase. The enzyme is pyruvate carboxylase. Pyruvate carboxylase. Carboxylase. Okay, so here the pyruvate is converted into the oxaloacetic acid, and in this enzyme ATP, biotin and CO2 is also utilized. ATP, biotin and CO2. ATP, biotin and CO2 is also used. Okay, so here the oxaloacetic acid is impermeable to the mitochondrial membrane, so it have to convert into the malate, okay, in the mitochondria itself. Okay, so in the mitochondria the oxaloacetic acid is converted into the malate, and malate is permeable. Malate is permeable for mitochondrial membrane, permeable for mitochondrial membrane. So membrane, MEMB, it's a membrane. Okay, so after converting the malate, after the formation of malate, it will reach towards the cytosol, and this malate will convert again into the oxaloacetic acid with the help of malate dehydrogenase. Okay, so and the enzyme is malate dehydrogenase. Okay, malate dehydrogenase, it will convert into the malate dehydrogenase. Malate is converted into the oxaloacetic acid again with the help of malate dehydrogenase. Okay, and that malate dehydrogenase conversion, during the malate dehydrogenase conversion, NADPH is formed. NADH is formed. Okay, now the oxaloacetic acid will convert into the phosphoenolpyruvate. Okay, phosphoenolpyruvate, and this phosphoenolpyruvate, in this the formation phosphoenolpyruvate conversion, the GDP is formed and the CO2 is released. CO2 is released and the GDP is formed. GDP is formed from the GTP and the CO2 is released. Okay, the phosphoenolpyruvate is converted into the 2-phosphoglycerate. 2-phosphoglycerate. Okay, 2-phosphoglycerate is generated. Okay, is synthesized, and the 2-phosphoglycerate is converted into the 3-phosphoglycerate. 2-phosphoglycerate is converted into the 3-phosphoglycerate. Okay, now the 3-phosphoglycerate, it is a reversible reactions. Okay, so here the 3-phosphoglycerate is converted into the 1,3-diphosphoglycerate. 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 Okay, 1,3-diphosphoglycerate is converted into the glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is converted into the fructose 1,6-diphosphate. Fructose 1,6-diphosphate. Okay, so here from the pyruvate, the fructose 1,6-diphosphate is synthesized. In the second step, the fructose 1,6-diphosphate is converted into the glucose 6-phosphate. Okay, so here in the second step, the formation of glucose 6-phosphate, from fructose 6-phosphate, fructose 1,6-diphosphate, fructose 1,6-diphosphate. phosphate. Okay, so fructose 1,6 diphosphate is converted into the fructose 6-phosphate with the help of fructose 1,6 diphosphatase. Okay, so fructose 1,6 diphosphate is converted into the fructose 6-phosphate with the help of fructose 1,6 diphosphatase enzyme. Phosphatase, and then fructose 6-phosphate is converted into the glucose 6-phosphate. Okay, glucose 6-phosphate. Fructose 6-phosphate is converted into the glucose 6-phosphate. Okay, here one is the cofactor, that is the Mg plus 2, magnesium plus 2 is also used. Now the fructose, now the glucose 6-phosphate is in the third step, the glucose 6-phosphate is converted into the glucose, okay, with the help of glucose 6-phosphatase enzyme. In the third step, the glucose 6-phosphate is converted into the glucose. Okay, so here the glucose 6-phosphate is converted into the glucose with the help of glucose 6-phosphatase. Okay, glucose 6-phosphatase. Okay, and the main characteristic of this enzyme, this is the mainly present in the liver and the kidney, but it is absent in the brain, in the muscles and the adipose tissue. So I will write here, it's a mainly present in your that liver and kidney, and absent in brain, muscles, adipose tissue. Okay, right. So here this is the main enzyme which we can say that in the last step conversion, this is the main enzyme which are responsible for the conversion of glucose 6-phosphate to the glucose. Okay, so this is the main reverse glycolysis which are the mainly involved for the glucose generation. Now we will discuss about the Cori cycle, means the glucose generation from the lactate, and we will discuss about the glucose generation from the glycerol and other glycogenic amino acid as well. Now come to the next gluconeogenesis procedure. Here the glucose is generated with the help of lactate, and this cycle is known as the Cori cycle. This cycle, I will make the separate video for the Cori cycle, but I am just giving the introduction about the Cori cycle. Here the glucose is generated with the help of lactate, and the enzyme which is used in this purpose, that is the lactate dehydrogenase. Lactate is converted into the pyruvate with the help of lactate dehydrogenase, and this pyruvate with the help of reverse glycolysis, which we have discussed before some time. Now the pyruvate is converted into the glucose with the help of reverse glycolysis procedure. Now in the third reaction, the synthesis of glucose from the propionyl coenzyme A. Basically the propionyl coenzyme A is the we can say it's a 3-carbon chain compound, and it is generated by the help of fatty acid metabolization, or we can say by the oxidation of fatty acid. Okay, by the oxidation of odd chain, it will generated by the oxidation of odd chain fatty acids. Okay, fatty acids. And some amino acids like methionine and the isoleucine also generated by the breakdown of methionine and the isoleucine also generated this propionyl coenzyme A. Okay, so this the propionyl coenzyme A is the with the help of propionyl coenzyme A carboxylase that convert into the methylmalonyl coenzyme A. This methylmalonyl coenzyme A with the help of mutase is convert into the succinyl coenzyme A, and this succinyl coenzyme A is the intermediate for the TCA cycle, tricarboxylic acid cycle, and it will convert into the oxaloacetic acid, or we can say oxaloacetate. Now this oxaloacetate is again convert into the phosphoenolpyruvate, which we have discussed yet here this is the phosphoenolpyruvate, and this phosphoenolpyruvate is convert into the glucose with the help of reversal of the glycolysis procedure. Now come to the synthesis of the glucose from the glycerol. The glycerol is generated in the adipose tissue by the triglycerides, by the metabolization of triglycerides. Okay, so the glycerol is converted into the glycerol 3-phosphate with the help of glycerol kinase, and this glycerol 3-phosphate is generated into the DHAP, dihydroxyacetone phosphate, is with the help of glycerol 3-phosphate dehydrogenase. This DHAP is converted into the fructose 1,6-diphosphate, and again with the help of reversal glycolysis procedure it will convert into the glucose. Now the fifth is the glycogenic amino acid. I have discussed just little bit about the glycogenic acid because it is not that much important. Only MCQ pattern point of view, it's a 1 or 2 questions they can ask. Okay, so here the most common amino acids which are involved for the synthesis of the glucose, that is the alanine. Different amino acids are metabolized and produce particular intermediates, and these intermediates are very much important for the gluconeogenesis. I have written here also the different amino acids produce intermediate for gluconeogenesis. Now some different amino acids are generated different intermediates. Now like this, alanine, serine, glycine and cysteine generated the pyruvate. Valine, methionine, isoleucine is converted into the succinyl coenzyme A. Glutamine, proline and histidine is converted into the alpha ketoglutaric acid. Asparagine and aspartate is converted into the oxaloacetic acid. So this is the complete gluconeogenesis. Now we will discuss about the significance of the gluconeogenesis. Now come to the significance part, because this gluconeogenesis help to maintain the glucose continuous supply. So the organs which are very much important, or we can say which needs the emergency supply, or we can say not emergency, it's a continuous supply for the glucose, because they are working 24/7. That's why the gluconeogenesis help for the continuous supply of glucose, like brain, like CNS, like RBCs, testes, kidneys, because these organs are continuously 24/7 hours working in the working mode. Okay, and if this in the skeletal muscles, as the anaerobic conditions are generated, if we are doing too much workout, so in the skeletal muscles anaerobic conditions are developed. So for during that anaerobic conditions, the glucose is supplied by the help of gluconeogenesis. Otherwise the too much lactate will deposit and it will harm our muscles. Okay, now during the fasting, if we are doing the fasting for a long time period, so the gluconeogenesis help to fulfill the body requirement for the glucose. Now the next significance, that with this cycle body remove the lactate, glycerol and the propionate. As we know that the lactate are the toxic for our muscles. So by the help of gluconeogenesis, our body help to remove such these toxic products, and with the help of these toxic products, our body utilize into the formation of the glucose. This is a whole for the gluconeogenesis. I hope you enjoyed my lecture. Any doubt, kindly ask in the comment section. Otherwise, please like, share and subscribe. Thank you.